Hi, this is Dr. Launen, and this is a lecture on bacterial horizontal gene transfer at the overview level. The goal here is for you to be able to define horizontal versus vertical gene transfer and to understand what horizontal gene transfer is at the overview level. First, let's start with vertical gene transfer. Vertical gene transfer is in fact what you're most familiar with. That's what humans do. So when humans reproduce and have babies, then the mother and the father are transferring their genes from parent to offspring. That is vertical gene transfer. In the prokaryotic world, when one bacterial cell or archaeal cell, shown here, goes through the process of binary fission to produce two presumably identical daughter cells, shown here on the right, then they are transferring genes from the original cell to the two offspring cells, and that is vertical gene transfer. And that's quite different from horizontal gene transfer. Horizontal gene transfer can be defined as the transfer of genes or genetic material from one cell to another that is not the offspring of the initial cell. So from donor cell to recipient cell, where the recipient cell is not the offspring of the donor cell. And there are three modes of horizontal gene transfer. They are transformation, conjugation, and transduction. This cartoon is an oversimplification in that it is showing two cells, one we're referring to as the donor and one we're referring to as the recipient. Now, these two cells are currently engaging in all three modes of horizontal gene transfer, and that wouldn't really happen in nature. In nature, what would happen is one or another would be occurring, not all three at the same time. So here are the three modes, however. Transformation means that the recipient cell is taking up naked DNA from the environment. The source of that naked DNA had to have been a previous donor cell that died and lysed and released its DNA into the environment. Recipient cells that are able to perform or to be transformed, to undergo transformation, have to be able to detect this naked DNA, attach to it, and then take it up and recombine it into their genomes in order for it to actually transform or genetically change the cell. This is a process discovered by Dr. Fred Griffiths in Streptococcus pneumoniae. Conjugation is a different form of horizontal gene transfer. In conjugation, the donor cell is able to chemically recognize a recipient cell. And the donor cell will then build something called a pilus. It's a protein bridge. It's an appendage, and it will connect the donor and the recipient cell. And once connected, the donor cell will be able to transfer genetic material into the recipient cell. The recipient cell will utilize that genetic material. It'll either form a little plasmid over here out of it, or it will recombine that genetic material into its own chromosome, thereby being genetically altered. So this form of horizontal gene transfer, referred to as conjugation, is also referred to as primitive mating, and it requires cell-cell contact. Transduction, the third form of horizontal gene transfer, requires a viral vector. The kind of viruses that do this are called bacteriophage. Here you just see the word phage written. And in this form of horizontal gene transfer, the virus, the bacteriophage or phage, is going to actually take DNA from the donor cell and carry it over to the recipient cell and inject it like as if it was a hypodermic syringe. In would come this DNA, and it would be utilized by the recipient cell as genetic material, thereby changing the recipient cell. So that's transduction. It does not require cell-cell contact, but it does require a viral vector. Those are the three forms of horizontal gene transfer. If you understand what I just said, and if you took appropriate notes, you should be able to answer the questions on the next slide. So here's a take 5 moment. You should complete this table, okay? So if this table had been blank, other than just these three things here, would you have been able to fill these in? So would you know that, these three forms of horizontal gene transfer, could you spell them? Spelling's very important. Would you know which one requires or doesn't require cell-cell contact, which involves the uptake of naked DNA, and which involves a viral vector? Would you be able to fill that table in? And if you can do that, it means that you're at just the very beginning of understanding horizontal gene transfer. You're starting to organize what is what and have a basic familiarity with the terms and concepts involved. You should also consider this question here. What does the term donor and recipient cell mean in the context of horizontal gene transfer? And how would you know that a cell was genetically altered? So let's consider the first. Okay, so what does the term donor cell mean, and what does the term recipient cell? The donor cell is the cell that is donating the DNA. It is transferring DNA from itself to another cell. The other cell that is accepting the DNA is referred to as the recipient cell. So back to these three modes of horizontal gene transfer. In the case of transformation, the donor cell is the one that died and released this naked DNA into the environment. And the recipient cell is the one that is being transformed and taking that DNA up from the environment. In the case of conjugation, the donor cell is the one that is able to detect the recipient cell, build a pilus, and then send DNA over into the recipient cell. In the case of transduction, here is the recipient cell, shown here, and it is gaining DNA through the phage vector, okay, the viral vector. The source of that DNA is referred to as the donor cell, and it could be this one over here, but it could be like a cell over here, somewhere else in the nearby environment. How would you know a cell was genetically altered? Well, there are only two ways to do that. One, you could sequence the genome and look for changes. And two, and this is more common, you could look for changes in phenotype. So if a cell that had been susceptible to antibiotics, then after a horizontal gene transfer event became resistant to antibiotics, that's a change in phenotype, and phenotypes can only be accomplished by changes in genotype. So that shows you that the cell was genetically altered through horizontal gene transfer. And that completes this introduction to the topic of horizontal gene transfer.